Yes, and a little bit of athletics too. You ready? Okay. Well, just want to welcome you all. We have some parents here, so welcome. Faculty, students. So a few years back, Ms. Liga had an idea of bringing some folks that have been doing things in the summer in front of the rest of you so you can see what they're doing. There's some really neat programs going on, and we have 3 people. We're going to originally have 4. One couldn't make it. She's out sick. But you're going to hear from 3 different people who did something pretty exciting over this summer. So maybe you'll find some connection that you think you might want to do this yourself, maybe next summer. So it's a great opportunity just when I get started. So first up, we have Jonathan Wolf Rosengarten, and he had the opportunity to intern with Massachusetts Audubon Society as an environmentalist. Working at Felix Neck Wildlife Sanctuary, Jonathan worked to promote conservation as well as our responsibility as human beings to protect our natural world. Without any further ado, Jonathan.
Alright, hey guys, so today I'm just going to be presenting to you quick, kind of looking into what I did this summer. Alright, so I interned in Martha's Vineyard, which is an island off the coast of Massachusetts. It's about 87 square miles and consists of 2 islands, Martha's Vineyard and Chappaquiddick. The vineyard is home to about 15,000 people year-round, and during the summer tourist months can reach population numbers of more than 100,000 people. It is one wildlife sanctuary named Felix Neck, which is a part of the Massachusetts Audubon. Felix Neck offers educational services, conservation programs, and wildlife excursions. It also plays host to walking and running trails used by naturalists and city dwellers alike. Alright, so what did I do? I developed field educational programs and led and assisted classroom and outdoor seminars focused on woodland and marine biology. I worked on curriculum used in their year-round educational services, summer camps, and festivals. Felix Neck is super unique in that it has an outdoor and indoor classroom. In addition to guiding classroom seminars, I also worked on identifying and documenting marine biology, invasive and native species to Martha's Vineyard. Probably the most interesting animal we were able to find and make a record of was the horseshoe crab. Horseshoe crabs are in the arachnid family and were alive and have not changed since the dinosaur age. Their blood is used in every single vaccination test because of the color and its ability to detect bacterial endotoxins. Some say that the death rate after they harvest their blood is 3%, but the mortality rate is actually closer to 30%. Horseshoe crab blood is very similar to human blood with the exception of the difference in colors and the fact that humans have iron, whereas horseshoe crabs use copper to carry oxygen through their blood. So in the second picture, what you're seeing is a horseshoe crab facility where they drain their blood. That is their actual color. And yeah, so they harvest them, and unfortunately the mortality rate is close to 30%, so they are kind of winding, they're trying to prevent that. So scientists have come up with a newer vaccination test that could replace horseshoe crab blood in the near future, but for right now we're still relying off it. Alright, so highlights. So I had the ability to write an article discussing the behaviors and lifestyles of diurnal, which are daytime, nocturnal, nighttime, and crepuscular, which are twilight animals on the island. The article was published in Felix Neck Environmental Scientific Newsletter. So being able to give back to communities such as Martha's Vineyard, which is overrun in summer with tourists who disregard environmental precautions, was incredibly rewarding. The environment needs to be protected and conserved, and it was super cool to be able to be a part of the solution to MV's problem of local expansion. The environment provides us all with so much, but is sometimes forgotten until it is beyond salvageable. Being part of a team that helped repair and renew the habitat and wilderness of the area was an enriching experience I will not soon forget. It was also very rewarding to be able to teach and provide educational services to family who visited the sanctuary. Helping them understand their surroundings and nature is something that they all benefited from, so it was cool to be able to watch the message reach people from all over. So necessity for nature and conservation. So as I have stressed throughout this entire presentation, overdevelopment and industrialization is a serious matter as it continually encroaches on our ever-shrinking natural spaces. Martha's Vineyard almost acts as a microcosm for the earth as a whole, and that it started off as an island, home to Native Americans and whaling captains who had a very light footprint on the land. However, as technology progresses and industry grows, nature has taken a hit. This is visible everywhere, as increased populations has a direct relationship with expansion into areas we need to protect. Now, I'm not saying that technology and industry are not beneficial. They certainly are. They help our economy grow, provide jobs, and increase money circulation throughout society. However, letting industry encroach on nature at an unchecked rate is the problem we must find a way to fix. This is the problem I was helping solve this past summer. There's no easy solution to this, but the efforts must be made. The internship acts as a catalyst for me to be able to be a part of the solution to this ever-growing problem. Thank you.
Very good. Thank you, John. Next up, Holden Neer. Holden had the opportunity to attend UCLA's Applications of Nanoscience Technology program. Here he was able to explore important applications of nanoscience, learn the basics of reviewing existing scientific literature, and go through the process of proposing a research project in pursuit of new scientific data. Holden.
Everyone. So most of you probably don't know me, but as Mr. Gardner just said, my name is Holden Neer, and I'm a sophomore here at the Benjamin School. So today I would like to talk to you about what I did this summer. Now I did something a little bit different from what everybody else did. I actually did a program at UCLA at the UCLA Summer Institute. So what is this institute? Now the institute is a collection of summer programs at UCLA on different subjects. There are several different subjects in the arts and sciences, so really this is applicable to everybody. Now, the program that I did, and we'll be talking with you about today, is the Applications of Nanoscience program. Now, at UCLA, there is a Nanotechnology Institute, and certain teachers and grad students who research the subject created this program. Now, it's a relatively new program, and it's only been open for a few years, meaning that they're still willing to change and listen to students for advice. Now, some of the great things about this program is, first of all, you do get college credit for it, and also, it's open to all high school students who have an interest in science. So, I know I've continuously been saying the word like nanoscience and nanotechnology, but what does the word nano really mean? It's actually pretty simple. It's just science on the nanoscale. Now, nano means 10 to the minus 9th, which means 1/billionth of a meter. So you all know approximately how long a meter it is. Now, take 1,000th of that. Now, you're still looking at a millimeter, which you can use, which you can measure on a meter stick. Now take 1,000,000th of that. Now you're looking at the microscale. And a person can still see with their own eye something that's 50 micrometers long. Now, as I said before, to get to nanometers, you have to take 1/billionth of that. That's over 50,000 times smaller than the smallest thing that you can see. Now, since we're working with things so small, many of the chemical laws that you learn in your science classes today don't apply to these substances. And that's one of the reasons why this study is so important. So what did I do at the program? So the program was 2 weeks long, and in the first week I got to learn about nanoparticles and how they're actually used in our everyday lives. This includes topics such as hydrophobicity, which is a surface's ability to repel water, as you can see in the top image, filtration of nanoparticles, and biosensors, which detect biological components in substances. So you may think that nanotechnology is something that's pretty far away from your life and you may never use, but that's actually quite untrue. Now you've probably seen companies or commercials that sell sprays that cause water to bounce off of things that would normally absorb it. This is actually caused by nanotechnology, and companies such as Neverwet use nanoscience to do that. As well as that, nanotechnology is being used in all new types of filtration. As all the water that you use today has to be filtered somehow, nanotechnology is beginning to make that easier. Now, the second week in the program, I actually did a research project in a group. So in groups, we came up with our own research projects, and we worked in a lab and did many experiments to find data for that project. Afterwards, we presented those results in front of several UCLA professors and researchers. So what was this project that we did? We created a synthetic skin to be receptive to pressure by passing a current through nanoparticles. So to make the synthetic skin, we needed something that was rubbery and that's quite like skin. So we decided to use something called PDMS, which is like a silicone rubber material. Now to do this, we put together a liquid base and a curing agent, which is special because both of those are liquids. But when put together and heated, it turns into the silicone solid rubber material. As well as that, we added iron oxide nanocrystals into that to make it conductive. And you can see the final result. It was our conductive skin. So on the left is a picture of the actual synthetic skin that we created, and on the right there's a cartoon diagram, and I'm going to use that to explain what really our synthetic skin was. So I've talked about what synthetic skin is and what it's made of, but now I'm going to explain how it actually works. So on the right you can see a zoomed in image of the synthetic skin. Now the black dots are the iron oxide nanocrystals that I mentioned before, and those are conductive, which cause the PDMS to be conductive. Now the current flows in between the red and black terminals of the multimeter, because electrons can jump from each particle to another particle, or as you can see here, from dot to dot. So you may be wondering, how does this cause the material to sense pressure? Well, when no pressure is being applied, it's relatively difficult for the electrons to jump from one place to another. This is because the particles are so far apart. Now, as you can see here, when it's being squeezed, the particles get closer together, and that means that it's easier for the electrons to jump from one place to another, and that makes the material more conductive. Now, you may not be able to visualize this, so just to help you better visualize it, imagine lily pads in a lake, and little frogs trying to jump from one lily pad to the next. Now, if those are relatively far apart, it's really hard for the frogs to get from one place to another. But if you bring them closer together, it's much easier and much more efficient for them to be able to get across. Now, this is exactly like the nanoparticles, because if the nanoparticles are closer together, it makes it much easier for the electrons to jump from one place to another, making the substance more conductive. So we want to test just how well our skin could detect pressure. So to do this, we measured the resistance of our synthetic skin when we squeezed the skin and when we released it. And resistance is basically the opposite of conductivity. Now, as you can see in the video on the right, the resistance was relatively high when no pressure was being applied. But when pressure was applied, the resistance dramatically decreased, meaning the conductivity increased. So, this was the graph that we made from our data, and note the low resistance corresponds with higher electrical conductivity, so the electrons can move more easily. So the 4 plateaus in the graph, which are the high points, is when no resistance was being applied. Now, those are the highest points in the graph because it was harder for the electrons to get from one place to another, which made the resistance higher. Now, as you can see, the resistance nearly always returned to the same spot, which was because no pressure was being applied, and that was constant. So the green, purple, and orange highlighted sections are where the resistance is when pressure is applied. Now, the most resistance was at the orange section, and that's where the most pressure was applied. The least resistance was in the orange section when the most pressure was applied, and the most resistance was at the low pressure, which is the green one. So you can see as more pressure is applied, the resistance continuously decreases. Thus, the synthetic skin can differentiate in between different amounts of pressure. So you may be wondering, why does this synthetic skin matter? Because we use things like skin grafts today if a person needs replacement for their skin, or if they are a burn victim or something similar. Well, if we can create a synthetic skin like this that can detect and sense pressure, we can return touch to the sense of people like amputees by putting the synthetic skin on prosthetic limbs. Now, synthetic skin does not have the same risk of infection, and there are reduced risks in many other places, and it's easier to put on. Alright, so the synthetic skin was able to distinguish in between varied amounts of pressure. Now, take into account that we were only able to work on this for 1 week, so imagine if we had much longer. In the future, this synthetic skin may be able to be connected to the user's nervous system, allowing them to recognize instantaneously where they're feeling pressure. As well as that, we want to be able to create the skin so that it can sense other stimuli, including temperature, vibrations, and allowing the user with synthetic skin to feel almost identically to how they would with real skin. Alright, so you may be thinking, well, this program is mostly being in a classroom and a lab for basically the whole time. But it's actually quite untrue. You actually get to do quite a bit of stuff. First of all, you get to stay in dormitories and basically live the college life and experience that. As well as that, you almost immediately make friends. Like the day I got there, I made tons of friends, which you can keep even after the program is over. Also, you have tons of free time. After each class or each lab, you can basically do whatever you want with your time, just like you would be able to in college. And during weekends, you can pretty much go everywhere because you have the weekends free, like Santa Monica, Hollywood, Beverly Hills. Those are just a few of the places that we went. And one of the most important things, if you guys are interested in science, is that at this program you're able to make connections. Now these connections can help you in the future, and can be with some of the grad students or researchers who you're presenting to or working with you. All right, so thank you guys so much for listening, and if any of you guys want to talk to me about it,
next up, Robert Rubin. He had the opportunity to intern in Dr. Thomas Bannister's lab within the Department of Molecular Medicine at Scripps Research Institute. The Department of Molecular Medicine is a research unit aimed at drug discovery and managed by chemists and biologists with extensive experience in the pharmaceutical industry. Their cancer projects targeted unique metabolic phenotypes of tumor cells, defining molecular characteristics to be exploited for the development of targeted therapies. Robert, come on down.
All right, so as you heard, I spent this past summer in the Bannister Lab at Scripps, and in the Bannister Lab, we basically try to make drugs that help, but good drugs, and we try to cure some diseases. The drug I was working on was trying to cure cancer, and I'm interested in telling you guys about it. So about the internship as a whole, it involves working 40 hours in a lab. It sounds like a lot, but you get really interested in the science, and it feels cool having like an official job. And it involved 10 students from all over Palm Beach County, and it involved like a 2-hour class on Tuesdays. And the interesting part about this is that I really wanted to do this, and I applied, and I got rejected. But what I ended up doing is that I sent some emails out to a lot of the doctors at Scripps, about like 10 of them in total, and Dr. Bannister said, I have enough room in my lab to take you in as a part-time intern. And so I showed up for orientation with an intern that was only at like 20 hours a week, wasn't a part of this official program, and I met the program director, and she said that I was rejected because there wasn't enough room, and since I sent all those emails out, and I was able to find room for myself, she took me into the main program. So if you're gonna take one lesson away from this, I would say that you should just really put yourself out there. If you get rejected from something at first, send some emails, try to make some connections, and good things will happen if you keep on trying. And so other than that, the best way I can explain my internship is by comparing it to my favorite TV show of all time, Breaking Bad. And so in Breaking Bad, there's a master chemist, Heisenberg, and there's his junior apprentice, Jesse. And their job is to make as much of methamphetamine as they can. And in my lab, I worked with a master chemist, Lee Padiza. He went to grad school at Scripps. He's originally from Zimbabwe, and he was a really cool guy to work with. He's incredibly smart. And I was his apprentice, just like Jesse was for Walt. And our job was to make as much of this compound as we could. So Breaking Bad, it took them all that trouble just to make that simple compound, and what I was making down there was a bit more confusing. So it took a lot of work to put into it. Okay, so when you're making drugs, or trying to find a good drug, you have to screen a lot of compounds to see which one is good, and it has a potential future. So there's actually a machine at Scripps called a high throughput screen that can screen upwards of 250,000 chemicals in an 8-hour workday. So it puts them against like a biological assay to see which ones could have a future as a drug. And then it gets narrowed down to about like 250. And where the drug I was working on was just before clinical trials. It is being used now for animal trials, and that's actually why we had to make it. It's because we sent it over to Tampa to be used in an animal study. But this does have a possible future as a drug, but as you can see, it take a lot of time. So this whole process is very lengthy. And when you're making cancer drugs, you want to see what's unique about cancer cells so that you can target them specifically. The current chemotherapies that we have are very good at killing cancer cells. The only issue is that they're also good at killing normal, healthy cells. So people who are undergoing chemotherapy also have negative side effects associated with it. So if you're able to find something that's unique about cancer, and something that a drug would only target specifically, it can kill cancer cells without killing normal cells. And that's where the progress needs to be made. And so one of the most unique aspects of cancer cells is the Warburg effect. So the Warburg effect states that cancer cells go through increased rates of glycolysis, and they go through lactic acid fermentation instead of going through oxidative phosphorylation. So that may sound very confusing, but what it basically means is that instead of taking the more efficient pathway that most of us take, it goes through lactic acid fermentation, which healthy people would only do unless they're going through extreme exercise. So pretty much most of the athletes here at school would produce lactic acid in some way if they get a cramp or something. Or maybe all of them except for the bowling team. And then so lactic acid is something that cancer cells produce a lot of. And if we study what happens to lactic acid, we can take advantage of it. So in cancer cells, lactic acid is usually transferred through the cell by the monocarboxylate transporter, which is a transport protein, brings the acid outside of the cell, and as a result, the pH stays constant and the cells are allowed to proliferate. However, if we create a drug that blocks lactic acid inside of the cell, it would block the MCT, and as a result, the hydrogen ions build up inside of the cell, the pH decreases significantly, the enzymes stop working, and eventually the cell dies. So if we are able to block the monocarboxylate transporter, we can selectively kill cancer cells without killing normal cells. So this brings us back to our molecule right here. This is a MCT1 inhibitor. So what that means is that it inhibits that monocarboxylate transporter. There are actually two different types of MCTs in the human body. There's 1 and 4, and some other drugs are being developed as MCT4 inhibitors, but this drug right here is being developed as a MCT1 inhibitor. It was originally developed by a pharmaceutical company called AstraZeneca, and the reason that I needed to make this compound is because, as I said, it's being used in an animal study to see if it has a future as a drug. And the reason why they can't just use it already is because, as you can see, it kind of has a complex structure, and it's not available in mass production. And one quick thing that I probably should mention is that when you're looking at these diagrams, these are how molecules are expressed in organic chemistry. So whenever you see like a point, but there's no letter next to it, that means it's a carbon atom, because organic chemistry is based mainly off the carbon atom. And so you'll see a lot of these diagrams coming up. Okay, so now that you've seen our molecule, I'll tell you how we went about making it. And so it may look complicated, but you can break it up into its component parts. The same way you can kind of make a Lego house out of individual Legos. So you may not be able to buy the house immediately. You can only buy the individual Lego parts. So we have many of these compounds you see to the right available in the lab. The average chemistry lab at Scripps has over 30,000 chemicals. So we have many of these available, many different Lego blocks available at our disposal. And the way we go about making it is that we build up, we keep on adding more Lego blocks onto it, keep on adding more compounds onto the main scaffold until we eventually get our final product. Oh, and that thing up there, you may think it means the Benjamin School, but it actually stands for terbutylmethylsilane. You'll see that a lot of these chemicals have really weird names, and all of them smell really bad. That's just an interesting fact. Chemistry labs smell pretty awful. And so this is what it all looked like, but don't get too overwhelmed. The important things to notice here is that there are 10 separate arrows, so each one of these reactions, each arrow represents a reaction, and it took 10 of them to get to our final product, which is right here. And you can see that as the reactions go on, the scaffold keeps on getting bigger and bigger as we're adding more of those Lego blocks onto it. And as we get to the end, we eventually were able to produce our final compound, which just looks like a plain white powder. It's pretty simple. We only produced 1 gram of it. And one other thing is that this whole process only took 2.5 weeks. So this is just one of the projects that I worked on at Scripps. So we actually went through this whole synthesis twice, and we were working on another project. But just as a general time frame, it took about 2.5 weeks to go through all of these 10 steps. The average reaction took about maybe like 6 hours. And the average yield, which is the amount you get based on what's mathematically possible, is around like 70% on average. And so how did we go about performing these reactions? This is what I think is the really cool part, because all of the machines that they have on Scripps are very advanced, and there are a lot of cool stuff I was able to use. So many of our reactions are very plain. They occur in a round bottom flask like that. But others, so we used a microwave reactor, which is actually pretty simple to what you'd use in your house. But it's used for chemical reactions, and it's used for ones at high temperature. And another mechanism we used is a reflux reactor, which looks like that. And so the gathering reactants part is actually pretty hard. As I said earlier, there's 30,000 chemicals that we have at our disposal. So we often had to go on a scavenger hunt looking for chemicals, which is pretty fun, but took a lot of time. And so once you have the reaction done, or once you have it going, you need to see when it's done. So you do that by taking a small sample, only 10 microliters, and placing it into this machine right here. And so this machine tells you how many different compounds are in there, based on how many peaks there are in here, and what the masses are for each of those peaks. And so by looking at this allows you to tell if the main compound that you have in your reaction mixture is the correct one. And if it is, that means you're done. So once the reaction's done, the next step is extracting it. So what this basically does is that you take your reaction mixture, put it into one of these separator funnels, and you add an organic solvent, which is something like ethyl acetate. And then you put water in there. Water is an inorganic solvent. And so just like when you add like oil and vinegar, they don't mix. These two don't mix. And so they form 2 separate layers. And all the good stuff we want, like our product, goes to the organic layer. And some of the salt byproducts will go to the aqueous layer, which is down here. So this allows us to eliminate some of our impurities before we proceed with the purification. So you purify it by placing our sample into a column chromatography machine. And so what this does is that it separates each different aspect of our compound based on their relative polarities. So each compound has a different polarity, and when you pass it through this column, the least polar stuff comes out first, and the most polar stuff comes out at the end. So you collect it at like 10 milliliters a time into, you'll set up a rack of about like 100 test tubes. You collect 10 milliliters in each one, and you'll have a range of them that has your target product in there. So after the purification, you make sure that your compound is the correct one by bringing it downstairs to the NMR machine. This is actually the same technology as MRI. And what it does is that it uses a large magnet to determine how many hydrogen atoms are in your compound and where they're located. And so what it basically does is that it creates a signature for your compound. Each different compound has a different NMR signature. So this is what one of them would look like over here. And by looking at the signature of our compound and the signature of what it should look like, if they're the same, then that means that we had the correct amount. And what this also tells us is the percent purity. In order to submit our compound, we needed to have over 95% purity. And this tells us how pure our sample is. And so after we made it, we sent it over to Tampa in the Moffitt Cancer Center, and now it's being used in an animal trial. It's being put into mice, and if it kills the cancer in the mice, then move on to the next step, which would be a clinical trial. So this is still a long ways down the road from being an official drug, but it's part of the research process, and it was really cool to be a part of it. And so I would like to thank everyone at Scripps, specifically my mentor, Lee, my boss, Dr. Bannister, and the program director, Rosie. And here at TBS, I'd like to thank Dr. Haley for inspiring my love for chemistry, and Ms. Saliga for organizing this whole presentation. And I'll leave you guys with the gift of a gummy bear being oxidized. This is a fun reaction we did on my last day there. Thank you guys very much.
Bowling yesterday had a wicked cramp. Unfortunately, Cameron Perosi is ill and unable to present today. Her internship at Max Planck Research Institute focused on neuroscience. Her presentation, Discovering the Memory Process in the Hippocampus Using Virtual Reality and a Mini BCS Board, is forthcoming, so stay tuned. You'll hear from her later. And Ryan Smith will be up now for the next presentation. So from academics to athletics. All right, give us